Bertinetti,
Yeah.
Resculpting the Visual Narrative, Robust Phenotypes for Variable Light Habitats. All right, cool. Take it away.
All right, so thanks everyone for being here. Thanks also to the organizers for inviting me. Originally, my advisor was supposed to present, so if someone was here for that talk, I'm sorry for that. I'll try not to disappoint you. So today I want to talk about how my previous preconceptions or maybe bias about visual ecology kind of influence how I went into a new project. So the idea is like basically when we look at visual systems, we assume certain things, or I used to assume certain things that change when I apply those questions to a different system, to a different set of organisms. So vertebrate eyes are fascinating. I don't think I need to tell that here. And specifically, I'm biased towards fish. I'm going to talk mostly about fish. I think the early conception was that, eyes are these sophisticated things. So people used to say quotes as everything in the vertebrate eye means something. One way that meant like, it's probably under strong selection pressure. So there's anything that you see there, that phenotypic variation might potentially have a meaning. At the same time, it meant that it was maybe not that likely to find differences across populations because, any slight change is going to have an impact of how this complex system works. So later on, we got to, some great work by Endler, which I think many people have approached in this symposium many times. My own interpretation of this, of course, with fish is that you have a signal, which in this case could be like social signaling for like conspecifics or looking for a prey. And then you have a receiver. And the environment is kind of what governs, especially in aquatic environments, which is some great examples before, how this is going to impact how this communication is happening. So, for instance, we could think if you want to increase, you want to stand out in your environment, then certain wavelengths that reflect, depending on the light that it's available, are going to be predominant, whereas if you want to be cryptic, maybe you should go for a different strategy. And again, that is highly influenced by which environment this is happening. Without getting into the complicated details about the background and so on, it's a simplified way of thinking of it. The key message is like you could change your signal. And of course, as we all know, and we just saw some great examples again, receivers can also change. It's a complex trait. So there are multiple levels at which this could be changing. And of course, if you want to make it more complicated, you could ask, okay, well, if you have a different species with a different set of signaling space probably and different systems, what is happening? So kind of when I started working in visual ecology, I was working with cichlid fish, again, kind of the to-go organisms where you're thinking about sensory drive or sensory ecology. And all these papers I read, all these studies were always like, okay, a lot of variation across habitats, even if you do get a depth within a single lake, you're going to see variation in the expression of certain opsins, certain genes, parallel evolution, rapid evolution, you name it. It was beautiful. I said like, okay, I can keep this doing forever. Go catch some fish and find the pattern. However, I moved to the US, so I thought it was great to maybe look at the different system. Centracids are a beautiful system, a small adaptive radiation native to North America, about 40 extant species, depending who you ask. And then popular game fish, right? So bluegill, largemouth bass, some of those are the fish that everybody knows. And more interesting, there's quite some good knowledge about their ecological roles. So basically, they have different feeding ecologies. So, that could pose a good system to try to find, again, can we disentangle light environments from maybe other sources that might drive evolution of their vision ecology. More interesting, even for me, is that people have claimed that these two groups, so basically cichlid fish and centracids, were to some extent convergent in many ways. So it was like, great, I just need to study these guys and find a lot of phenotypic variation. So the study was that we selected a subset of species that occur at Wisconsin, where we study those, and then we could go and look at two lakes, right? So we have our dark lake, our clear lake, and then just basically ask what are the phenotypic responses that we are seeing. It could be that, as we would expect, maybe all fish, normally if they move to clear water, they will have an enhanced sensitivity to blue parts of the spectrum. Or, it could be that every species, given that they have their own ecological niche, are reacting differently. So that was one of the main questions that drove this study. So first of all, what is the environment? So these are kind of some pretty videos of how this looks like. Right away, you can see that in the dark lake, the distance at which you see the fish is like, a greater distance. And then also that the amount of colors that you can see, there's just more details in this clear water lake. Of course, this is a human view perspective, but just wanted to give you an impression of how they look like. If you actually want to measure this, again, irradiance, the word in the room, I guess. So it's the amount of light that it's allocated throughout the visible spectrum. And if you compare the blue line, which would be our clear lake, to the dark lake, which is our orange lake, these are 1 meter depth. So you can see that the amount of light, it's kind of shifted towards this orange-red side of the spectrum. And not only that, but also the amount of light that it's available, it's highly reduced. So this is like great. I want to roughly mention that these lakes are kind of far away and there's no river connecting them or anything like that. And then, if we look throughout the depth profile, basically, this could be only a small spot of the lake, but, is this a property of the water body? We actually find that that's the case. So what you're seeing here is just every dot representing the photon distribution at every single depth as you go down. And then we can see that in the dark lake, it basically becomes less intense and more turbid. And the opposite holds true for the clear lake. At the same time, at every single depth that you measure, there's more light available in the clear lake than the dark lake. So that was a long story just to say, okay, we do have our contrasting habitats that should impose some selective pressures on the system. So what if we look at the receiver? Of course, there are many things that could be changing. One could be morphology. It could be the lens transmittance, so basically the amount of wavelengths that even make it to the retina. And then inside the photoreceptors, we know that there are visual pigments that somehow account for a lot of the color vision. There's been so many talks, I don't know if I should get into the details, but basically you have an opsin protein plus a chromophore, and that will somehow determine the peak sensitivity of those pigments to different light. Basically, you can say there's a red, a green, or a blue pigment based on that. Okay, so results. First of all, the morphology part of it. So again, the subset of our simplified phylogenetic tree for those species. And then when we saw, first of all, when we look at relative eye width or lens diameter, which are kind of two ways of measuring spatial resolution or visual acuity, we do see a significant effect of the species. We don't see any effect of the environment. That was kind of interesting, but we also look at lens transmittance, and it seems like this is kind of something that's restricted to this family at least. So here we don't even see differences among species. It's kind of set at that 400-something nanometers. Okay, but then, of course, we could look at the gene expression for looking for those opsin proteins and the chromophore. So we had some RNA-seq data. Now I'm showing the phylogenetic tree again. First is kind of the relative abundance of cones to rods. So it seems like all these species seem to have more rhodopsin expression, which is used for dim light instead of color vision. But once again, only the effect of the species, no environmental component. And when we look at the relative expression of the opsins, first of all, we see that it's morely a dichromatic system. So it's all the bars seem like green and red. And then we don't see strong differences across environment. So it was kind of a bummer. We can also look at CYP27C1, which is just a gene that is involved in the exchange of those chromophores. So it has some role also in switching the sensitivity of the visual pigments. Once again, we see a difference among species, but not the environment. And this is not what I thought when I went into this. One of the hypotheses is that if we look at the retinal structure of those photoreceptors in centracids, normally this is the kind of pattern that you will see throughout the retina. So the bigger cell, so within the red circle, the bigger cells are double cones, and the one in the middle is a single cone, and that's kind of repeated throughout the retina. And if we now look at what the RNA-seq data was telling us, and genes that are in the genome, those are the kind of 4 genes that we found, but then actually expressed in adults, and I should emphasize the adult part of it, are only these 2. And then some additional data suggests that greens are mostly only in the single cones, red are only in the double cones, so that might explain why there's not much to be changed there. So it's a pretty restricted retina in terms of which pigments can be expressed in different cells. So what about the signal? Maybe the receiver is not changing, but of course we have the component of the signal. So for this, we measure 5 points. Reflectance data also was introduced today, so I don't go into the details. But basically what we did is a PCA. So all the measurements for one individual, we plotted all that data into a PCA. And then this is what you basically see here in the x-axis is PC1, y-axis PC2. This is about 90% of the variation, so a significant amount. And then, for example, for one species, the upper one, that's a bluegill, you could see that that's the space, the reflectance space that they're using within those two PCs, right? And this was already more interesting because if you compare those responses to the pumpkin seed, which is the fish below, they are kind of switching the space that they're using depending on the environment. So that's already pointing at some sort of environmental component. You could expand this to different species that we had, still see some cool patterns. So it's like, cool, at least there's some variation showing up. So if you take that PC1 as the absolute brightness, so to say, that you could measure in those, we do now see G/E interaction. So some species are enhancing their values in that PC1, which is kind of difficult to define, but I'm happy to talk about that. And then some others are actually decreasing it. More interesting, also intraspecific variation seems to vary a lot, and I think there were some talks touching upon that. We also see a G/E interaction here, and that's the centroid distance. So that's basically if you think about every individual in that reflectance space, how far away it's from the center of the cloud. You could take that as a measure of the variation within that cloud, right? And actually, the two that are actually different are the second column, rock bust, and the last one. And those are actually showing higher variation in clear environments. So I attribute that to the fact that in broader, clear water, you have more spectral range available. So maybe they are able to use a broader reflectance space. Okay, so conclusions, upstream expression as highly variable in aquatic environments. I think that was my to-go phrase for the last presentations I gave on cichlids always. In this case, it's not universal, so I should not assume that, and maybe other people in the field shouldn't. Species-specific responses in body coloration, that's what we found. I think that's really interesting. It means that, again, when we think about visual ecology, most of the time it has been focused on sensory systems. But I think there's the signaling component that only by changing that you might get enough variation in the system. And then broader reflectance space in broader environments. I kind of touched upon that a bit. And then foraging performance might not rely on color vision. Red and green system seems to do well for this fish. They are pretty abundant. And it might be that once you lost that spectral complexity, those genes, then you have a constrained visual system that it's not easy to regain. So for that, I think I'm a bit over time. Sorry for that. But I would like to thank many, many people that are involved in this project. Thanks.